All right, good morning, everyone. I'm Nick Gregg. I'm one of the epileptologists here and do research on neuromodulation. Happy to fill in with moderating this morning for our first full speaker session titled Intraoperative Mapping and Biomarker Discovery. The first speaker I'll introduce is very near and dear to me, Dr. Klassen. He trained me for 2 years of my DBS fellowship. All around wonderful guy, has a background in computer science, technically very skilled, avid ham radio operator. If you ever have a question about a drone, go to Bryan. Fantastic videos. But in any case, excited to hear his talk this morning about DBS, Intraoperative Electrophysiology, and novel mapping.
Thanks a lot. All right. So it's great to start off the session this morning. This is my first slide, and you're probably wondering what that is. So I'll give you a clue. It's a single green P, right? And you're probably wondering why I'm showing you this. Well, it's a useful size reference for the deep brain stimulation targets that we're asking our surgeons to implant in movement disorders. So the subthalamic nucleus and globus pallidus interna, these are the targets for Parkinson's disease. The VIM nucleus of the thalamus tends to be where we go if there's a tremor disorder. And the volumes are on the order of the size of a P. In fact, if you look at the motor subregions of these nuclei, which are the true targets, the comparison's more apt. And within the motor regions, there's probably symptom-specific regions. So in the subthalamic nucleus, which is already tiny, you probably have a different area for a tremor-predominant patient that you're targeting than you would for, say, an akinetic rigid patient. So these are very tiny structures in the order of a P or smaller. We're asking our surgical colleagues to take a DBS electrode that's about 40% the width of a single strand of uncooked spaghetti and skewer that pea-sized region in the middle of the skull without direct visualization. So that's the order of the challenge, and I think this is a good picture for me because as a neurologist, when I'm looking during planning or when I'm looking at postoperative lead construction, these are 3 of our patients, we're looking at this at extreme levels of magnification, so it's easy to lose sight of what's actually happening. All right, so what I'm going to do in the next few minutes is tell you how we're currently clinically use neurophysiology to help our surgical colleagues ensure that we're implanting these targets as best as we can. And some of this has been, I guess, previewed by Dr. Miller because there's a lot of parallels between the BCI work and then what we're doing deeper in the brain with these structures. So in this case, we have to have a target that's defined based on imaging and reflected here in the thalamus as that red star. And then the surgeon, with a frame system, you heard about one such system from Dr. Lee, but will set up a cannula or tube that points toward the target. And that's the tube through which the permanently implantable electrode will ultimately be placed, but you do have the opportunity to place a microelectrode probe first, okay? And then there's a micro drive, can be controlled by the surgeon or can be controlled electronically, to advance this probe in sub-millimeter increments, so very precisely down into the target. Along the way, from the business end of that electrode, we'll get a voltage trace. So you can record the electrical potential difference between the microelectrode tip and a reference. We use the cannula. And if we zoom up on this, as Dr. Miller showed yesterday, we'll see a fair amount of content. You have very low frequency information. This is a few seconds. That's probably pulse artifact, all the way down to this spiky activity. Now what we do in intraoperative monitoring is we filter. We take all of the activity under 300 hertz and we throw it out. We then are able to look at the single units, and you can see that threshold in red anytime the black trace crosses. That's probably a single unit represented of a single action potential from one neuron. And this is how it looks in the OR. We have a clinical system, and in this example, at about 8 millimeters above the target, we have this highly filtered activity. You can see a few units. Any trace that crosses that blue line will get rastered over to the side, and you can start to see how many units are there at once. You can start to see the shape of the units, okay? So what does this tell us? How do we use this? Well, we can do a couple of things with it. First, we are able to define borders between structures with a high degree of spatial accuracy. So particularly between white matter and gray matter, I'm going to show you an example of Dr. Miller driving one of these probes down through white matter into the subthalamic nucleus. And you'll be able to hear a difference, and you'll be able to see on the upper right-hand corner of this screen, our computer screen, and you'll be able to see a change there. So this is sort of white matter baseline. And with movement, there's a little sound. But now you start to hear this change, and you can see the units there on the screen. Okay, so with sub-millimeter precision, we're able to define that border, which isn't really something you can necessarily even do on imaging because there can be blooming artifacts, et cetera. The other thing we can do is delineate patient-specific motor representation. This may be a little different, even in the same structure, even if two patients have a similar appearing thalamus, the patient-specific representation may not align. Here's an example of a patient moving. Every time he moves his hand on his own, you can hear the associated firing. Okay, so we know that that area is associated with his movement. And the other thing we can do is look at sensory representation. And in this next example, it's a different depth, same patient. Instead of having the patient move, I'm passively moving his fingers, activating the muscle spindles, which feeds into the thalamus, and we can hear that activity. And this cell was interesting because it didn't fire, it only fired with extension, not flexion, right? So you have to kind of do a broad sampling to kind of see if there's a unit like that there. And that's interesting because this is a kinesthetic cell. This is the marker for the VIM target, basically. The other thing we can sometimes see is correlates of the disease state, particularly in tremor disorders. So I'm going to show you a cell that's firing in synchrony with this patient's tremor as we're monitoring that tremor with a wearable accelerometer and EMG. So I hope that's coming through okay. But that rhythmic activity, that's not normal activity. That's because this patient has a tremor disorder. All right, so this is all very interesting. I'm going to show you now the most common pattern probably that we observe during microelectrode recording. Okay, so this is a little less interesting. We got maybe one token unit there. But the fundamental problem is a similar problem to what Dr. Miller described with BCI, is single unit recordings are a fundamentally unstable biomarker. Some of that comes down to the small region of tissue that we're actually sampling with the tip. And we're also qualitatively interpreting this data, so that requires multiple individuals with a high level expertise working in tandem to really use this. And then that, therefore, represents a fundamental problem with scalability in care as we're trying to deploy these technologies into psychiatric conditions and other type of conditions. So over the next few minutes, the rest of my talk, I'd like to talk about our vision for how we're going to change this and hopefully improve it for our patients. First, we need to start applying quantitative analyses to these signals. We want to use a broader range of signal features. We want to move beyond just single units. There's a lot else out there. And then we want to do recordings in parallel so that we can analyze activity at multiple different brain regions simultaneously. So this is an example, again, of units that we do here now with I guess, our technology 1.0, as people have been calling it, on our way down to the GPI target. And we can see, or really listen, is what we're actually doing clinically, to the firing patterns. Okay? That's semi-quantitative, the expert analysis. But we're working, particularly Dr. Inche's team, working to statistically look at these patterns and see if we can quantitate the timing of the spikes. But the other thing that we're interested in is this morphology or the shape of the spikes. That's been something that's not really been looked into too well. And we've been able to use novel AI algorithms developed by Dr. Miller for actually evoked potential analysis, which you'll hear about more later. We've been able to use those in our intraoperative microelectrode recordings. So here's how it works. You can see on the top the raw voltage trace, right? And then this algorithm is able to decompose. We can sort the spikes, and this algorithm can decompose the spikes into a series of canonical shapes. And that is done naively, so we don't have to say this is the shape that we're looking for, or we're looking for 5 different shapes. The algorithm will just handle that for us. There'll be a scaling factor, so if the shape's a little bigger or smaller, we can still lump them together. And then you can remove those and do analysis on the residual activity without the spikes if you'd like. So we just published this technique, and as a part of that technical publication, we showed this example of one of our patients where we recorded down into the GPI from multiple depths, multiple different units, and the algorithm was able to sort these, again, naively into 6 different canonical shapes. And they did tend to cluster spatially. So for example, this particular shape was found primarily within our GPI target. Now, the next step, of course, is to apply this across multiple patients, and we're hoping that there are canonical shapes that might mark GPI not just from this patient, but across patients. And this would be another tool in addition to analysis of the timing that will allow us to quantitate this and kind of spread this out to wider patient use. However, I also said we want to move beyond just single cell action potentials, and that's partly because there's a lot of information, as I mentioned, we're throwing out that probably is useful, and also this spatial sampling issue with that small high impedance microelectrode, we're going to get a wider range of activity sampled from larger electrodes. And we're able to do that on the microelectrode. There's a ring electrode just above it that we can get the local field potential or the EEG of the deep brain. Dr. Miller talked about how you can see oscillations or rhythms at a specific frequency, and then broadband change, which is more of an overall activity underlying the electrode. And so we've done this. This is a recording from one of our patients in the thalamus. You take the time series, you do a Fourier transform, you get it into the frequency domain, and this is just a resting state of what's happening in that person's brain. But we also had this patient do a task where he'd move his hand or he'd rest. We did it several times and averaged that all together. Now you can compare the spectra during movement and during rest. And so this mirrors what has been seen in the cortex. In the low frequency oscillation range, you know there are oscillations because there's the peaks there. There's less power, there's desynchronization of these oscillations to some degree. There's a little frequency shift too. And that's been shown before in Thalamus, but what we were the first people to really describe, what we were the first to really describe in detail is that we can see in many of our patients this broadband shift. Okay, that's known in the cortex, but we've now seen it in the thalamus during movement. And this is an average of all that over time. You can look in the temporal, with a little more temporal precision and see that the broadband starts at movement and kind of cuts off right at the end of movement. Now, if we repeat that experiment in multiple locations in the same patient, you can start to develop a map. You can do a cross-correlation to quantify that, and then you can plot that back on the patient's own anatomy. And so what we see is this decrease in the oscillation, this low-frequency oscillation, is pretty widespread. We saw it everywhere we recorded in the thalamus, whereas the increase in the broadband activity was spatially discrete, probably toward our target. And indeed, when we look at this across all of our patients, we saw that the increase in broadband activity was localized to motor thalamus, and that's the VIM nucleus and the VOP nucleus of the thalamus, whereas the low-frequency oscillatory changes were more widespread. We were a little disappointed because we were hoping to resolve this border between the VIM target and the VOP, and it doesn't seem like this simple technique can really do that. It's an important border because the VIM is the cerebellar input zone to the thalamus, and it's our therapeutic target. Whereas ahead of it is the VOP, where it's getting basal ganglionic input, and you may get some tremor control there, but it's going to be suboptimal. So we'd like to figure out how we can find this border, and we were hoping that there were more subtle features in the signals that our data, or that our simple analyses couldn't really uncover. And of course, in the age of AI, has been talked about a lot, structure within our data can be learned by algorithms. And so we've teamed up with folks at the Technical University of Berlin machine learning group that's led by Klaus Mueller, who's also one of the co-founders of the AI Agnostics, sort of AI medical company, and looked at data from our patients. So we've taken recordings from multiple patients who've been implanted in the thalamus, sequentially recorded the local field potentials, the time domain data there, and then we run a Fourier transform to move it into frequency space. And then we've developed and applied novel deep learning algorithms to then be able to classify additional patients. And this is a typical pattern that we've seen, and it's pretty exciting to us because, first of all, we've been able to have the algorithm cluster the recording sites into, say, the yellow, which is the internal capsule. This is a border between gray and white that probably isn't surprising that we found, and is visible on MRI. But the most exciting thing is we're seeing in all of the patients an intrathalamic border or transition zone, which we believe is probably the transition between VOP and then our therapeutic target VIM. And the other thing that's exciting, and this is not visible on MRI, and the other thing that's exciting about it is we don't need a task to uncover this. This is all in resting data from our patients. So this is ready for publication, but we're waiting for permission from our intellectual property people before we get that out. And then I want to get back to this idea of serial versus parallel recordings. So clinically, and all the research projects I just went over have used data where we record from a site, we do what we're going to do, we move down, we record again, and we keep repeating that. But we want to move toward parallel recording, where we will put a probe in or a stylet through that cannula that has recording surfaces that basically span the entire range that we're already looking at. Now we can record simultaneously, and the advantages are pretty, it'll be very advantageous. So first of all, it'll be more efficient use of the OR time. Now, whether that's just clinically, we can put the probe in, we can run all these algorithms and get our answer very quickly. But also for research, one of the key barriers to this type of research is the amount of time it takes in the OR, and of course you can't prolong OR time that much. So you're getting a very little bit of data from each patient. And for example, that map I showed you, we had to repeat this kind of open-close task that maybe takes a minute. We had to repeat that 14 times for all those sites. If we had this, we could just have them do that once, and you have a map. The other thing is the maps we do make are going to be a little better, so we'll have more detailed neurophysiologic topography, and that's because we have now electrodes that are radially arranged around the surface of the probe, and so we'll be able to get a little better idea of radial orientation rather than just up and down the electrode. And finally, we'll be able to do what we absolutely can't do when you're recording at separate spots at separate times. We can look at simultaneous interactions between different sites in the brain. Coherence is one of the signal processing metrics to do that, and there's others. So this is what we're trying to move toward. We were fortunate enough to get money through a Minnesota State initiative to design and then develop a novel electrode that we're hoping would allow us to do this, and then put it through preclinical testing. And so we've done that, and we're able to prove that we can record these data. This is the local field potential. We can also record some unit activity. And of course, you can subject that to frequency analysis, and then kind of a proof of concept. We can find regions of coherence between different areas along the span. And so now what we're doing is working with industry partners to try to bring this technology into human use, initially obviously for research, but ideally the vision would be we place this probe in, we do the sorts of analyses that I talked about in my earlier slides, and we can get a really accurate map very quickly of if this is a good site to implant and where along that trajectory, and then potentially even use that information back in the neurology clinic to help us set a program that's going to really target that zone. So this is what I had. And again, our vision is basically to move the future of intraoperative DBS neurophysiology to focus more on quantitative analysis. We're far behind, say, where they're at in the fMRI realm with this so far, at least clinically speaking. And we want to look at a broad range of signal features, and we would like to do that from multiple brain regions simultaneously. And because of the scale of this whole problem, interpretation is going to have to be assisted with artificial intelligence. We think that we at Mayo are in a strong position to pioneer these efforts because we have excellent clinical expertise. We have high patient volumes. Our patients are really invested. They're pretty happy to participate in these experiments usually. And then we have a core group of people who have quantitative toolkits that are skilled in signal processing. And a lot of these same people are also the clinical domain experts. And then we've made some key collaborations. So I think with all of that and with the extra sort of push from the BIONIC Initiative, we'll be able to push this forward. And that's what I had. Thanks for listening.